My name is Lai Thai Leong and my metric number is 196891. So today I'm going to talk about what is microarray and what is the applications of microarray. So before we can understand what is microarray, we have to understand a few terminologies first, or words. Okay, so what are DNA, genes, and genomes? DNA is actually known as the oxyribonucleic acid. It is the genetic materials of all organisms. So even us, the human, also contains DNAs in our cells, specifically in the nucleus. Okay, so actually DNA is made out of letters that encodes the information that is needed by an organism, including human, to survive. So there are actually four biological letters that write DNA, which are adenine A, guanine G, cytosine C, and thymine T. So these letters are known as nucleotides. So actually, DNA can be thought as a long stretch of sentences or words that convey meaningful information. For example, it can be TAA, T or TATAAT, something like that. And for genes actually are words. They're made out of different biological letters or nucleotides. So actually genes is like a recipe used to make different products, different foods, something like that. Okay, so for the genes, the product will be proteins. So if our mom cooks, then the product will be our food. So for the genome is actually a sum of all DNA in an organisms, which includes all its genes. So genomes is like a cookbook. If the gene is recipe, genome is the cookbook. It's a cookbook which not only contains the recipes, which is genes, but also contains non-recipes like front pages, index. So for the genomes, this kind of front pages or index may be the promoter, something like that. And then what are alleles and polymorphism, and also SNV. So alleles are actually different versions of the same gene. So this is similar to the different spellings in the Americans and also British English, like program without the ME at the front, at the back, and also program with ME at the back, which is the British English. So the stakes of having different forms or versions of the genes is known as polymorphisms. So poly basically means many and morph means forms. So actually it's many forms. So for the SNV, it is actually the abbreviations of the single nucleotide polymorphisms. So it's different in only one nucleotide. Okay, so when the gene is only different in only one nucleotide for different alleles, for example, C become G, and then this is known as SNB. So for our daily life, we can see that the British English and the American English is different, like analyzed with the Z and analyzed with S. You can understand in this way. So for indels and SNV, these two are also important words that we need to understand. So indels actually means insertions, deletions. Insertions is the additions of nucleotides. For example, for this sequence GCAGC, there is an insertion or additions of the letter T. Now it becomes GCTAGC. So, for example, our color undergoes insertions of one letter U. Then it becomes COLOUR, color. So it means the same things, it is the same genes, but there's one more extra letter. So for the deletions, it's the reverse. There's a loss of nucleotides. For example, GCAGC becomes the reverse. So for the color, it becomes of one U becomes COLOR. So for the copy number variations, there are actually different numbers of copies of repetitive sequence. For example, the CAGs can repeat 3 times. And at the same time, it may repeat 5 times, like the one at the right side. So, for example, inverts. American versions have only 1 L, and then the British versions have 2 L's. So there's variations in the number of letters. what are mRNAs and proteins? So mRNA is messenger RNAs. Messenger means it will convey the message. So it's a temporary copies of information used to form the proteins actually. The sum of all these mRNAs in the cells or in the tissues will be known as transcriptomes. And then the proteins, which is the workers of the cells, perform different functions like to digest our food, to protect us against the microorganisms so that we wouldn't fall sick, and other functions. So this is the flow of biological informations. From genes, which is the recipes, we can copy it in a process known as transcriptions to become an mRNA, which is a temporary notes or copies. And then it will be translated into proteins, and then our product will be made. So actually mRNAs can be reverse transcribed in a process known as reverse transcriptions. Basically it means that the notes that were copies become to the page in the code book, the recipes. Reverse process occurs. So when the genes under transcriptions to form the mRNAs, it is undergoing gene expressions. There are two types of gene expressions, which is upregulated and downregulated expressions. So up and downs are different, right? So different expressions. So differential expressions. So differential expressions can occur in different cells, in different conditions, or in different samples. So I will talk about it later. And then now our focus is DNA microarray. So this is the pictures of DNA microarray. You can see that it's very beautiful, right? So what is actually DNA microarrays? So what is it? Okay, according to the Cambridge Dictionary, micros mean very, very small. And then array means a large number of products of similar types. So the DNA microarrays is actually a glass-like spotted with many DNA to test for the presence of target DNA. So basically on the same slides, there are many spots that can test for different DNA. So imagine now I have to draw 16 circles within the rectangles. So what will happen? On the left side, the circle is very big. So I need 8 rectangles to draw 16 circles in total. But if I made it smaller in the upper right sides, there's only 2 rectangles needed. If I make the circle even smaller, only 1 rectangle will be enough. So we can say that the smaller the circles, the less the rectangles needed. So similarly, what if this circles is actually the spot, the dot, on the DNA microarray, which is the slides that I showed you just now? So when the spot is very big, we need 8 slides of microarrays. If the spot becomes smaller, we only need 2. And if the spot is even smaller, then we only need 1 slide to test for all 16 different DNA in the microarray slides. So the smaller the dots, which is also can be known as the spots, the less the slides needed to test for the same amount of DNA. This is the concept for microarrays. That's why the spots are very small. So maybe you will ask, how can we perform the microarray analysis? So this is the steps. So initially, we can divide it into condition or treatment 1 and also condition or treatment 2. Okay, so the left side is treatment 1, the right side is treatment 2. The cells and tissues under condition 1 and condition 2 are incubated in different tubes. And then the RNA extractions will be carried out. Means that we extract the message, and then from this RNA we reverse transcribe. We undergo the reverse transcription, as I mentioned just now, to form the cDNA, which is actually DNA. After I get the cDNA from both condition 1 and condition 2, I will mix both of them. Okay, then I will incubate them and wash off the unbound cDNA. After I incubate the cDNA with the DNA microarray, I will wash off the unbound cDNA. Then I will visualize or see the bound cDNA using a scanner, something like a camera but not camera. And then we will retrieve and analyze the merged or the combined visualizations of the gene expressions. What are the applications of microarrays? So the applications of microarrays are actually to detect a single nucleotide polymorphisms, we call it SNPs, to detect the copy number variations, and also to detect the indels. It can also help us to discover new genes. These functions are very important so that we can differentiate different strains of the same species. For example, maybe different strains of hibiscus. We have pink hibiscus, we have red hibiscus, and so on, and also to study the effect of drugs, what happens if we eat Panadol, and to study the effect of toxins, what if I got food poisoning, what will happen in our genes expressions, and also to study the differential expressions of gene, which is what I mentioned just now. Some genes will be upregulated, some genes will be downregulated. So this differential expression is like you will need different recipes when you encounter different conditions. When you want to cook curry chickens, then you want to search for the recipe to boil the chicken plus how to make the curry paste. Then you combine them. These two recipes is needed for the cooking of curry chickens. But when you want to bake maybe chocolate cakes, then you need the recipes to make how to make chocolates and another recipe to bake a cake, and then these two different recipes combinations is needed for different foods. So how to study the differential expression of genes? By using microarrays, this can be done. Last but not least, disease analysis can also be done by microarrays, so we can determine whether this person is sick or healthy using this microarray. So merged visualizations of gene expressions will comes out with this kind of results. We have red results and also green results, and this is the final products. We have green, red, and also yellows. Then the genes 1, 3, 4, 5 is in red, gene 4 and 7 is in green, and also gene 2 and 8 is in yellows. This is what we can see based on these results. Okay, so maybe condition 1 is normal, condition 2 is tumor, and then yellows means that genes is expressed in both conditions. So treatment 1 is control or treatment 2 is toxins. Then we know that when we didn't eat something toxic, then this group of genes will be expressed. And if we maybe get food poisonings, different set of genes will be expressed, maybe to help us to remove this toxin so that we can become healthy again. From this microarray, we can know a lot of different things. So I think it will be clear right now. That's all. Thank you.